Okay, this unit focuses on classical Mendelian genetics. That is the sort of science of inheritance that was first determined, elucidated by Gregor Mendel. Now, Mendel, as you've heard plenty of times, you'll read about and you'll hear about in this class, in your college genetics class, he's widely considered as the father of modern genetics. Now, he wasn't the first one to note that traits sometimes skip a generation, but he carefully designed and executed experiments that included numerical and statistical data rather than what a lot of biologists were doing at the time, which is just using sort of descriptive information. And he developed his own laws, postulates, principles of segregation and independent assortment, which we're going to learn about in just a bit. His model organism was the pea, and the cool thing about the pea was that it came in varieties, and each of these varieties had sort of alternate forms of traits. Here we see purple flowered plants and white flowered plants, but you also have round and wrinkled peas. We'll talk about the biochemical basis for that in just a bit. Now, the pea plant was actually an exceptionally good plant to use for the sort of experiments that he did, because true breeding varieties had been identified. These are varieties that when you breed them amongst themselves, they generate the same variety again. Peas are simple to cultivate. They have a short generation time, so you can do lots of experiments. You can eat. They have easy to manipulate gametes, as we'll see in just a bit. And if you don't want to fool with the gametes, they can self-pollinate, in which case that turns out to be useful too. So how can you manipulate the gametes? Alright, well, first you need to emasculate the plant that you're going to be using as the female plant. As you know, flowers have both male and female structures in them, so you got to get the male structures out of there so they can't contaminate the existing female structure. So you cut off the stamens, and let's zoom in on that to take a closer look at this flower. So there we go. Let's try that one more time. Okay, there we go. All right, so the pollen, which is the male gamete, is basically presented on these structures called the anthers, which are riding on stalks called the filaments, which is collectively called the stamen. What typically happens is when pollen is moved to the stigma here, there is a pollen tube that grows down a structure called the style into the ovary, where it fertilizes the female gametes. And now at this point, what you can do is you can transfer pollen from another plant to the first plant that you have emasculated, and the pollen will grow down, or grow a tube down into the ovary, fertilize the ovules. At this point, you can cover the first one up with a bag to prevent contamination, etc. Now, when those ovules, which are now fertilized and grow up into seeds or peas, they'll grow into this seed pod here, and all of these individuals here are the result of a separate fertilization event. They're brothers and sisters, more like fraternal twins, definitely not like identical twins, and each of them grew up to be members of the next generation. And so what happens in the cross between two true breeding individuals is that all the offspring have purple flowers. So let's look at the result of these, or let's look at this cross in a little bit more detail. Here we have the first generation, the P generation. That stands for parental. They're the parents, and you have purple flowers crossed with white flowers. Now, in the F1 generation, which we'll call the F1 generation because it stands for philia, which means brother, in that generation, aka the hybrid generation, all the plants have purple flowers. So then if you take the plants from the F1 generation and you allow them to self-pollinate or self-fertilize, or cross with each other, in the F2 generation you get this kind of interesting result. You get now white-flowered plants again. So it appears that this trait has skipped a generation, but you don't get half and half. In fact, you get about a 3-to-1 ratio of purple-flowered plants to white-flowered plants. And one thing that Mendel discovered when he did this experiment while monitoring other traits of these plants is that it just wasn't in the purple versus white, purple times white situation that he got 3-to-1 ratios in the F2 generation. We can take a look at axial flowered plants versus terminal flowered plants. That is, are the flowers on the ends versus somewhere in the middle. And he got 651 to 207 in the F2 generation, which is about 3.14 to 1 ratio. We've got yellow seeds by green seeds, true breeding for both in the P generation. You take it the F2, you get 6,000 to 2,000, about 3.01 to 1 ratio, and this is really close. Look and check out the numbers we've got here and the closeness of the ratio here. If you cross round seeds by wrinkled seeds in the parental generation, and then you get F1s, which are all round. Cross the F1s, you get in the F2s about a 3 to 1 ratio of round to wrinkled. With inflated pods to constricted seed pods in the F2 generation, you get a 2.95 to 1 ratio. See where I'm going with this? With green pods versus yellow pods, about a 2.82 to 1 ratio. From tall plants times dwarf plants, these are true breeding individuals in the parental generation. F1 all tall, F2 about a 2.84 to 1 ratio. Okay, you're seeing where I'm going with this. And Mendel noticed this too. He said to himself, gosh, there's this really strange ratio that keeps popping out. It can't be just because of random chance. There must be some principle. There must be some law of nature. There must be some rule that I can generate, some mathematics that I can develop, an understanding I can develop of this system to explain why time after time, trade after trade, this happens. And so he developed this whole system ex nihilo, from nothing. And what's amazing, it's without an understanding of chromosomes, without an understanding of DNA, without decent microscopes, and even though we'll talk about where his theories kind of fall short, but he's basically correct. That's what makes it an amazing intellectual achievement. Anyway, so let's describe this sort of system that he developed. It's basically there are a number of postulates that we'll cover that describes the behavior of what it is he saw that was operating behind the scenes. First of all, the first postulate was that there are unit factors that determine in sort of how traits manifest themselves, and these are genetic characteristics. We know them now as genes. He didn't use the word genes. And they exist in pairs in individual organisms. When each individual gets one of the unit factors from each parent. And these don't change throughout the generations. That is, the unit factors, they are inherited. And how they interact with each other kind of determines the expression of the phenotype. But the unit factors themselves don't change. And so that was an important idea as well. As I said, the unit factors are the genes. What we'll call the alternate versions of these unit factors is alleles. Now, an easy way to remember the term allele is it's got the AL in the front of it. So it's like alternate versions of genes are alleles. Not a different gene, just an alternate version of the same gene. It's sort of like, okay, you think about blue genes, right? You can have blue genes that are blue colored and genes that are white colored and genes that are tan colored. They're alternate versions of those kind of genes. Anyway, so the genes comprise something called the genotype. That's the genetic makeup of an individual. And the phenotype is what he saw, and it's really all he had access to, which was the external appearance, the traits, how they're manifest. Now, we know now that the reason that each individual has, two unit factors, two genes for every trait, is that genes are part of DNA. DNA is chromosomes, and there are two chromosomes that you have. They're called homologous chromosomes. You get one from your mother, one from your father. And the genes actually live sort of on the chromosomes, and you can have variations of those. And Mendel called those alleles big P and little p. And we'll see in a bit why we use the letters big P and little p. And here's why. His second postulate was a postulate of dominance and recessiveness. The idea is that when two unlike unit factors responsible for a single character are present in a single individual, one unit factor is dominant to the other, which is said to be recessive. One gene is expressed over the other. Now, the dominant allele will always be expressed in the phenotype, and the recessive allele will only be expressed when the dominant allele is not present. Now at this time I need to define some other terms. We've got homozygous. That means if you have the same allele for a particular trait on both of your chromosomes, of your homologous chromosomes at a particular location, you're homozygous, the same. Homo means same. Heterozygous means having different alleles for that trait on your chromosomes. Hetero means different. So let's take a look at the genotype and the phenotype, homozygosity versus heterozygosity, and how this kind of manifests in the purple and white situation. So let's take a closer look here at the genotypes. So if you have a homozygous dominant genotype, we'll call it dominant to distinguish it from recessive. It's big P, big P. It means you got two copies of the dominant allele, two copies of the alternative alleles for that particular gene. If you have one of each, you would be called heterozygous, and it doesn't matter whether it's big P, little p, or little p, big P, those are the same. And then if a plant has two versions of the recessive allele, it's noted with a genotype of little p, and you might call this homozygous recessive. Now the phenotypes that are generated from each of these genotypes is 3 purples and 1 white. In those are the ratios in offspring of potentially hundreds of plants. The idea here is that anytime you have at least one big P allele, you're going to have a purple flowered plant. We'll talk about the biochemistry of that in just a bit. Okay, let's consider Mendel's third postulate, the postulate of segregation. Segregation means separation. And the idea is that whenever plants are making gametes, which is pollen and ovule, or people are making gametes, egg and sperm, the paired unit factors separate randomly so that each gamete is likely to receive one or the other. And now that we have generated the term randomly, likelihood, you're probably thinking to yourself, okay, there's some probability involved. And indeed, there is. Let's follow what happens now, paying attention to the gametes in those first crosses. In the parental generation, you have each of the true reading individuals is homozygous, so each of them can only produce one type of gamete. The gametes have only one of the two alleles, either big P or little p. Put those two together, and now you have a heterozygote in the F1 generation, and that's why all those are purple. Alright, let's move on to the next cross. In the next cross, you have the F1s, each of which are heterozygous. Now, they generate gametes. Half the gametes are big P, carrying the dominant allele. Half the gametes are little p, carrying the recessive allele. Now, this diagram here that is used to explain how the gametes come together to generate the genotype of the offsprings was developed by Reginald Punnett. It wasn't used by Mendel, but it's really helpful in this case. So the ova or the ovules here could be of two different types, and the sperm or the pollen could be of two different types. When those come together, they'll come together in sort of equal proportions, is the theory, and these squares hold the offspring, or at least the ratios of the offspring, the ratios of the genotypes of the offspring. And you can see that there are three, or I should say, if you have a big P gamete coming together with a big P gamete, you get big P in the offspring. And there are two options where you can have a big P and a little p gamete coming together, and then one option when you can have two of the recessive gametes, or the gametes with the recessive alleles in them, coming together. And so that's how you get the 3-to-1 ratio in the F2 generation. Now, where does actually segregation occur, in the real world of chemistry and molecules and bonds, etc.? Segregation, that is, the sort of the splitting of the gametes, occurs in meiosis. We'll talk about that much more in the next chapter, but the idea is that whenever you're producing a sperm and egg, or pollen and ovule, if you're a plant, during anaphase 1, the chromosomes go in different directions, and they each carry an allele in those directions. Let's talk about the test cross. The test cross is how you can determine the genotype of an individual expressing the dominant phenotype. Because as you may realize, is that just because an individual is expressing the dominant trait, you don't necessarily know if they're homozygous dominant for that trait. Or if they're heterozygous. Okay, so the way that this works is, all right, here you have your individual that has a dominant phenotype but an unknown genotype. Are they big P, or little p? And what you can do here is you can actually cross them with the recessive phenotype. That has a known genotype, little p, and because it's recessive, it kind of hangs back, in the crosses and lets whatever kind of gamete is produced by this dominant phenotype be expressed in the next generation. Alright, so you can have a couple of different outcomes when you cross those. You could either have 100% purple in the next generation, or 50% purple and 50% white. You don't need to, sequence the DNA to determine what the genotype is of the mystery individual. The idea is that if they were homozygous dominant, big P, then all of the offspring would be purple, because that dominant allele would dominate over the two recessive alleles in the gametes of the homozygous recessive individual. And if you get a 50-50, then you know that it was heterozygous. One of each of the different types of gametes would be produced. When the dominant gametes were produced and they cross with either of the gametes from the other individual, you get purple flower plants, and the recessive gamete crosses to make white flowered plants. So that's how the test cross works. Alright, that's the end of this particular lecture series, and stay tuned in just a little while to we'll talk about dihybrid crosses. But for now, that's the end of that.